Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome back to the deep dive. Okay, here's a thought. What if the very roots of anxiety, like how our brains actually respond to stress, are laid down before we even take our first breath?
It's a pretty wild idea, isn't it?
Yeah, imagine our brain's vulnerability being shaped, while still in the womb. That's basically the premise of this groundbreaking study we're digging into today.
It really is a pivotal piece of research. For years, scientists have kind of suspected this link, right, between experiences in the womb and mental health later on. But the biological how, well, that's been mostly a mystery. So today we're dissecting this new preclinical study. It's from Weill Cornell Medicine, just published in Cell Reports September 10, 2025. And it's already making some waves in health news.
Right. So our mission today is really to unpack the intricate mechanisms behind this connection. We want to understand what these researchers actually found deep inside the brain.
And the implications, too.
Exactly. The implications for understanding anxiety and, maybe most importantly, for maternal health. So let's peel back the layers.
Let's do it. So the big picture problem, like you said, is that we've known for a while that difficulties during pregnancy, things like, say, major stress, infections, inflammation, they can increase a child's risk for psychiatric illness down the line.
Right. We've heard that connection before.
But pinning down the precise biological pathways, the exact steps that translate what a mother experiences into, her child's vulnerability later, that's been incredibly tricky.
So if what was kind of known, this study is finally tackling the how, what was their main question going in? Like, what were they looking for in those brain cells?
Exactly. Their goal was to find these persistent changes in brain cells, changes caused by let's call it a challenging gestational environment.
Okay.
And changes that lead to anxiety-like behavior later in life. To do this, they used a really clever approach, a mouse model.
A mouse model, right. How do you set that up in a lab to mimic that kind of environment?
Well, they induced abnormal immune activity in the pregnant mothers, basically creating an inflammatory environment during gestation.
Okay, like simulating a kind of stress or infection response.
Exactly. A controlled way to mimic that challenging prenatal experience. And here's the really crucial part. The offspring, the baby mice, they were genetically normal. Their genes weren't changed.
No.
The only difference was this exposure, this specific inflammatory environment while they were developing in the womb.
So identical genetics, different prenatal environment. What happened? Did these offspring show any signs later on?
Oh, absolutely. When these mice grew up, they showed classic signs of anxiety.
Like what?
Well, things like actively avoiding open, sort of unprotected spaces and showing a much bigger response to stressful situations. It looked a lot like what we see in human anxiety disorders. The lead researchers, Dr. Miklos Toth and Dr. Kristen Pyle, they actually call this setup the MIA model, maternal immune activation.
MIO model, got it. So they established the outcome, the anxious behavior. But the real deep dive, the core of this, is finding the mechanism. Where in the brain did they start looking?
Right. This is where it gets really precise. They zoomed in on this specific part of the brain called the ventral dentate gyrus, or VDG for short.
The VDG. Okay, where is that?
It's a key part of the hippocampus, the brain region involved in memory and emotion. The VDG, specifically, seems to be really important for assessing threats in the environment.
So it's like the brain's early warning system for danger.
That's a great way to put it, yeah.
Okay, an early warning system. That sounds critical. So what did they actually see happening in the VDG of these mice, the ones exposed to adversity?
Well, what they found was pretty striking. In these mice, a small but significant percentage of the neurons in the VDG became overly active when they faced a potentially threatening situation.
Overly active, not just a little bit more active.
No, significantly so. And this overactivity wasn't just some random finding. It was directly linked to those avoidance behaviors, that heightened anxiety. It's like their internal threat alarm was just set way too high permanently.
Wow. So it's a measurable physical change in how the brain perceives threat. But how does that happen? How does an experience before birth leave such a lasting imprint? What's the molecular secret?
This is where they made a really big leap. They looked at something called DNA methylation patterns.
Methylation. Okay, refresh my memory on that.
Yeah, so think of them like little chemical tags or sticky notes attached to our DNA. They don't change the actual genetic code, the sequence itself,
Right.
but they act like on-off switches, or maybe dimmer switches. They control which genes are active and which aren't. These are called epigenetic changes.
Epigenetics, meaning above the genome. So it's like instructions written on top of the genetic code,
Yeah.
telling the genes how to behave.
Exactly. Highlight this gene, mute that one. And these changes can be really lasting, even though the underlying DNA sequence is the same.
Okay, so what did they find about these epigenetic tags in the VDG of the anxious mice?
They found thousands of sites in the VDG where these DNA methylation patterns were altered compared to the control mice.
Thousands, wow.
Yeah, and what's really significant is that these changes were especially common in the regions of DNA that control synapses.
Synapses, the connections between brain cells where they talk to each other?
Precisely. So these epigenetic tags were essentially reprogramming how those conversations happen between neurons in this key threat detection area.
So the tags are kind of setting up a default response, like pre-programming certain neurons to freak out more easily.
That seems to be the case. As Dr. Kristen Pyle, one of the lead authors, put it, these epigenetic changes are instructing certain neurons in the VDG to respond differently in adulthood when faced with unsafe environments.
Instructing them.
Yeah. She explained that these specific neurons show too much activity, which makes the environment seem more threatening than it really is. It's like a hardwired, heightened sense of danger established before birth.
That's quite profound. The idea that the environment during pregnancy can fundamentally reprogram how an adult brain perceives danger.
Okay, but these are changes. How did they actually watch this happen? See the reprogramming in action? This was another really cool part of the study. They actually monitored these specific VDG neurons in real time, using advanced techniques while the mice were navigating a maze, moving between safe spots and areas that were potentially threatening, like open spaces.
Okay, so they're watching the brain cells fire as the mouse makes decisions. What do they see?
In the control mice, the normal ones, activity in the VDG was pretty steady, but in the mice that had the prenatal adversity exposure, something really remarkable happened.
Yeah.
Activity in these key neurons surged the moment they even started thinking about moving from a safe place into a threatening one.
Wait, before they moved, not as they encountered danger.
Exactly. It wasn't a reaction to the danger itself, but an anticipation of it.
Wow. That almost sounds like the definition of anxiety, doesn't it? That anticipatory dread.
It really does. They called it a ramping up of activity, and it seemed to encode this exaggerated perception of danger, which then drove the anxious behavior, the avoidance. Dr. Plow really emphasized this point. The increased activity is not a consequence of going into the threatening environment, but precedes it, suggesting the mice were anticipating danger.
That's such a powerful insight.
Yeah.
And if these neurons are being reprogrammed epigenetically, that must show up in their gene activity too, right? Did they look at gene expression?
They did, and they found that the VDG cells that were activated during this threatening task showed way more changes in gene activity, about 4 times more compared to the VDG cells that weren't activated.
Okay, so the ones doing the ramping up had their genes working differently.
Right, and many of these changes were in genes related to, guess what? Synapses, confirming that idea of altered communication pathways.
Makes sense. Were these gene changes linked just to anxiety, or did they see broader connections?
That's another important finding. A significant number of these altered genes are actually already known risk factors for other serious neuropsychiatric conditions,
Like what?
particularly schizophrenia and psychotic disorders, which lines up with other research suggesting maternal immune activation might be a risk factor for a spectrum of mental health issues, not just anxiety.
That connection is really sobering. Okay, so they looked at activity levels, epigenetics, gene expression. Did they also look at the sort of electrical nitty-gritty?
Yeah.
How the neurons were firing?
They did. They went even deeper into the electrophysiology. So imagine the brain has natural brakes, inhibitory signals to keep neuron activity in check.
Right, checks and balances.
Exactly. What they found was that in these adversity-exposed mice, those brakes in the VDG were significantly weaker. It's called disinhibition.
So the neurons are less controlled, more likely to get overly active.
Precisely. And to watch this directly, they used a special calcium biosensor, GCAMP6s, which lets you literally see neural activity levels light up.
And what did that show in real time?
It confirmed the earlier findings. In the mice exposed to adversity, the calcium signal indicating activity was significantly higher in the VDG when they were moving from safe to threatening areas, and crucially, it spiked before they fully entered the threatening zone.
Wow.
It just reinforces this idea of an enhanced perception of potential threat. The brakes are weak, and the accelerator is hypersensitive, especially to the idea of danger.
So these brains are essentially wired for high alert, anticipating threats because of changes that happened before birth.
Okay, stepping back. What does understanding this specific mechanism really mean for how we think about anxiety and what we can potentially do?
Yeah, this is where we get to the big implications.
Yeah.
This study gives us one of the clearest mechanistic links we've ever had between the prenatal environment, these specific epigenetic changes in the brain, and the later risk for psychiatric issues like anxiety.
It's like seeing the molecular blueprint for the first time.
Exactly.
And it really hammers home how critical maternal health during pregnancy is, doesn't it? It's clearly not just about physical outcomes like birth weight. Not at all. This shows it can profoundly shape long-term brain function, mental health. It's a much deeper impact than maybe we fully appreciated.
So could understanding these epigenetic signatures, as you called them, lead to practical tools, like ways to identify risk earlier?
That's the hope. A better understanding could potentially lead to early biomarkers for anxiety risk in people. Imagine being able to identify individuals who might be at higher risk before symptoms even show up.
That would be a total game changer, wouldn't it? Moving from just reacting to symptoms towards, well, maybe even prevention. How far off might that be?
It's definitely still early days for human application, but the concept is powerful. If we could reliably detect these specific epigenetic patterns, maybe through advanced brain imaging or eventually even less invasive methods, it could give us this unprecedented window.
A window for what? Like earlier support?
Exactly. Maybe targeted interventions during key developmental periods. It could really shift how we approach mental health care.
And what about treatments? For people already struggling with anxiety, especially maybe the kind that feels really ingrained, does this open new doors?
Potentially, yes. Those specific synapse-related genes that get abnormally programmed in these primed neurons, they could become targets for new therapies. Like Dr. Tooth said, understanding this mechanism might help explain that persistent stress sensitivity and avoidance you see in some people with sort of innate anxiety. If we know the specific faulty wiring, maybe we can eventually figure out how to gently nudge it back.
It's incredibly exciting research. But like all good science, we have to talk about the limitations too, right? What are the caveats here?
Absolutely. First and foremost, we need to remember this is a preclinical study. It used mouse models.
Right, not humans.
Exactly. While it's highly informative and a crucial first step, translating these findings directly to humans needs a lot more research. We're looking at fundamental principles here, but human brains and experiences are obviously more complex.
Okay. And you mentioned the epigenetics. They found changes in only, 10 to 30% of the cells? What does that tell us?
That's right. It highlights the complexity. It's not like every cell in the VDG is reprogrammed. It's a subset. So future studies using more advanced single-cell technologies are needed to really tease apart which specific cells are most affected and how those methylation changes directly link to gene expression changes in those specific cells.
So it's more nuanced than just flipping a single switch.
Definitely. The diversity of cells just within the VDG is significant, and understanding why certain neurons are more vulnerable to these epigenetic changes is a key next step.
And the brain's a network, right? Well, the VDG is clearly important here. Are other areas likely involved too?
That's an excellent point, and the researchers acknowledge it. While the VDG is a critical hub in this process, other parts of the hippocampus, like areas called CA1 and CA3, are definitely involved.
And they connect elsewhere.
Yeah, they connect to other major players in the brain's anxiety circuits, like the amygdala and parts of the prefrontal cortex, how those regions respond epigenetically, and how they all interact. That's another whole layer to explore. This study really opens the door to looking at the entire network through this new epigenetic lens.
Okay, so bringing it all together, this deep dive, really shows us something quite profound, doesn't it? That the environment in the womb, this really delicate period, can leave these lasting epigenetic marks on our brains.
Yeah, imprints that influence how we perceive and react to threats years down the line. It's this incredibly complex dance,
Oh.
Chemical tags, gene expression, neurons firing, all happening in this tiny brain region, setting the stage for our emotional lives.
It really makes you think. And it raises a provocative question, maybe for everyone listening. If the basic architecture of our threat response can be subtly changed before we're even born, What other parts of our personality, maybe our resilience, or even our predispositions?
Yeah.
What else might be quietly shaped by that earliest environment?
It's a huge question, and it leads to another one, doesn't it?
Which is,
What responsibilities does this understanding place on us, on society, to really support maternal health, not just for the physical well-being of the next generation?
but for their mental and emotional landscape too.
Yeah,
It really reframes the importance of prenatal care and support, doesn't it? Thinking about prevention in a whole new way.
absolutely. It highlights the incredibly deep, long-lasting ripple effect of providing care or not providing it during such a critical time.
Well, thank you for walking us through that complex research. Hopefully this has given everyone listening a richer, maybe more nuanced view of where anxiety might begin.
It's fascinating stuff.
It really is. Thanks for joining us on The Deep Dive. Until next time, keep digging deeper.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.